Module 2, Good Manufacturing Practice, Introduction to GMP APIs. About this module. Follow the prompts on the screen to navigate through the module. Quick quiz questions appear throughout the module. These are simple knowledge tests and are not scored or used in your final assessment. Final assessment at the end of the module is your opportunity to demonstrate your knowledge, and you will receive a score on these questions. All answers have appeared in the learning material. Navigation tip. Screens are interactive. Navigation prompts may occur at the top or bottom of the screen. Objective. The objective of this e-learning module is to provide an understanding of the GMP regulations relating to the manufacture of active pharmaceutical ingredients, API. The objectives of this training module for trainees is to learn what is needed for good manufacturing and quality management and assurance, understand regulations regarding personnel, premises, equipment, and utilities, understand equipment maintenance, cleaning, and calibration requirements, know the GMP regulations involved in API production, learn GMP processes for managing materials, packaging, storage, and distribution, identify the requirements for GMP in a laboratory setting, develop knowledge for validating products and processes in accordance with GMP, identify the steps involved in a product's rejection/rework/reprocess, understand how to apply GMP to contract manufacture. The guidelines of GMP for APIs. WHO Technical Report Series number 957, 2010, Annex 2. In this training course, we will cover each section of this annex, highlighting key concepts. Other guidances, e.g. ICH, Q7, and PIC's Guide to Good Manufacturing Practice for Medicinal Products, Part 2, are essentially the same with only small differences. Introduction to GMP Manufacturing. The GMP guidelines cover receipt of materials, production, packaging, repackaging, labeling and relabeling, quality control, QC, release, and storage and distribution of active pharmaceutical ingredients, API. Definition. The word manufacturing in GMP refers to all aspects of manufacturing, including storage, warehousing, laboratory, record keeping, clinical trials. Scope of GMP. The scope of the GMP for active pharmaceutical ingredients and those of most regulatory authorities includes both human and veterinary drug products. Importance of regulations. The manufacture of active pharmaceutical ingredients is now strictly regulated by the authorities due to a number of incidents over the years, where the root cause was found to be the API manufacturer's poor GMP. A sterile injectable product, heparin, was suspected of causing 246 deaths in the U.S. in 2007 and 2008. The root cause for these deaths was traced back to the API plant. Role of the quality unit. Quality management. The code details the responsibilities of production and quality staff. These two functions should be headed by two independent people. These two functions are required to be independent and have different reporting lines in order to avoid conflicts of interest and undue influence of production on QA decisions. Role of the quality unit. Continued. Who is responsible for quality? Everyone is responsible for quality. Quality cannot be tested into the product. It starts at product development, the design and qualification of the facility and equipment, and in the process validation. It continues with ongoing manufacture and statistical monitoring of product yield and quality. Deviations, trends, or adverse events should be thoroughly investigated, and the root cause of the problem should be identified, evaluated, and eliminated to ensure quality products. All staff should be observant and report any deviation or unusual occurrence during the manufacturing process. Role of the quality unit. Continued. The quality unit. The quality unit fulfills both quality assurance and quality control responsibilities. They are responsible and have the legal responsibility to sign off on the final release or rejection of all raw materials and product. These units should be independent of production. No materials or products should be released or used until this unit has completed their evaluation and the materials or products have been deemed satisfactory for release or use. Usually, the responsibility resides with the head of quality. However, this is commonly delegated to other people within the quality unit. Delegation must be done in writing. All the staff with delegated responsibilities should have the appropriate education, experience, and training for that role. The quality units should review and approve all appropriate quality-related documents. Role of the quality unit. Continued. These responsibilities should be described in writing and should include, but not necessarily limited to, 1. Releasing or rejecting raw material, intermediates, and API. 2. Establishing a system to release or reject raw materials, intermediates, packaging, and labeling materials. 3. Reviewing completed batch production and laboratory control records of critical process steps before the release of API for distribution. 4. Making sure that critical deviations are investigated and resolved. 5. Approving all specifications and master production instructions. 6. Approving all procedures impacting the quality of intermediates or APIs. 7. Making sure that internal audits, self-inspections, are performed. 8. Approving the intermediate and API of contract manufacturers. 9. Approving changes that potentially impact quality of intermediates or APIs. Role of the quality unit. Continued. These responsibilities should be described in writing and should include, but not necessarily limited to, 10. Reviewing and approving validation protocols and reports. 11. Making sure that quality-related complaints are investigated and resolved. 12. Making sure that effective systems are used for maintaining and calibrating critical equipment. 13. Making sure that materials are appropriately tested and the results are reported. 14. Making sure that there is stability data to support retest or expiry dates and storage conditions on APIs and or intermediates where appropriate. 15. Performing product quality reviews. Role of the quality unit. Continued. Internal audits. It is a GMP requirement to formally perform self-audits to proactively identify potential sources which may affect product quality and to ensure that issues are addressed in timely and in effective manner. The internal audit should be performed as per predetermined and approved schedule, and the internal team should be represented by all the department. The audit finding and corrective action should be documented and brought to the attention of senior management. The company must be able to show evidence that there is an internal audit, self-inspection program in place. Personnel. Introduction. The GMP rules recognize how critical people are to ensuring product quality. People. Quality assurance systems and adherence to GMP procedures relies exclusively on people. Responsibilities. There should be detailed and up-to-date position descriptions for everyone with manufacturing responsibility to ensure GMP compliance. Qualifications. There should be a detailed resume, copy of education certification, and training records for all key personnel. Hygiene. Good hygiene, good sanitation, and health habits should be practiced. Clean clothing and appropriate protective clothing should be worn at all times. If you have open sores or any infectious disease, you should report this to your supervisor. Consultants. Consultants advising on the manufacture and control of intermediates or APIs should have sufficient education, training, and experience, or any combination thereof, to advise on the subject for which they are retained. Contamination. Smoking, eating, drinking, chewing, and the storage of food should be restricted to certain designated areas separate from the manufacturing areas. Food and personal medicines should never be taken into the manufacturing areas. Buildings and facilities. Introduction. Buildings and facilities used in the manufacture of intermediates and APIs should be located, designed, and constructed to facilitate cleaning, maintenance, and operations as appropriate to the type and stage of manufacture. Contamination. Facilities should be designed to minimize potential contamination. Where microbiological specifications have been established for the intermediate or API, facilities should be designed to limit exposure to objectionable microbiological contaminants as appropriate. Special clauses apply if the product is a sterile or aseptically processed product. Segregation. There should be segregation between products, equipment, and materials to prevent mix-ups and contamination. Control. There should be defined areas or other control systems for the following activities: receipt, identification, sampling, and quarantine of incoming materials pending release or rejection. Quarantine before release or rejection of intermediates and APIs. Sampling of intermediates and APIs. Holding of rejected materials before further disposition, for example, return, reprocessing, or destruction. Storage of released materials. Production operations. Packaging and labeling operations. Utilities. Introduction. All utilities that could impact on product quality have to be qualified and monitored appropriately, including, but not limited to, compressed air, nitrogen blankets, purified water, factory air. Procedures. Written procedures should be established assigning responsibility for facility and utility maintenance, sanitation, and cleaning, as well as the use of suitable rodenticides, insecticides, fungicides, fumigating agents, cleaning and sanitizing agents. Pipe work. Pipe work must be correctly identified and should be designed and used in a manner to avoid contamination. Buildings. Buildings used in the manufacture of intermediates and APIs should be properly maintained and kept in a clean condition. Water. Water must be of a suitable standard for its intended use. As a minimum, World Health Organization potable water should be used. However, to assure API quality, a higher standard may be required. Water treatment systems should be qualified and monitored. Containment. A. Appropriate measures should be established and implemented to prevent cross-contamination from personnel, materials, etc. B. Dedicated production areas, i.e. facilities, air handling equipment, and/or process equipment, should be employed in the production of highly sensitizing materials such as penicillins or cephalosporins. C. All highly potent materials, e.g. cytotoxics, sex hormones, etc., should be segregated and handled in dedicated production areas. D. Any production activities, including weighing, milling, or packaging of highly toxic non-pharmaceutical materials such as herbicides and pesticides, should not be conducted using the buildings and/or equipment being used for the production of APIs. Handling and storage of these highly toxic non-pharmaceutical materials should be separate from APIs. Process equipment. Introduction. The GMP guidelines ensure that process equipment is designed to minimize product contamination and mix-ups. Fit for purpose. Equipment should be of an appropriate design and size, be suitably located for its intended use, and have a qualified operating range, i.e. the equipment should be qualified. Equipment should be constructed so product contact surfaces do not alter the quality of the product during manufacture, and where possible, closed/contained systems or equipment should be used. Maintenance and cleaning. Equipment should be suitably maintained and cleaned. Validation protocols should exist for the cleaning with acceptance criteria for residues, cleaning procedures, and cleaning agents. Calibration. All measurement equipment should be calibrated, and the calibration should be traceable to national standards and should be identified to indicate the calibration status. Computerized systems. The diversity, complexity, and criticality of your computerized systems will determine the extent of validation required and the risk that computerized system poses to the product quality and patient safety. Using the risk framework defined in the ISPE GAMP5 guidelines, non-configurable, commercial off-the-shelf COTS, computerized systems, GAMP Category 3 may require less validation efforts than bespoke systems, GAMP Category 5. Protection of your computerized system and data security are key requirements, so business continuation, backup, and disaster recovery procedures and validation evidence is essential. GAMP5 is used as a guideline only. Tip 1. Never use someone else's password or give your password to someone else. Tip 2. Always lock your workstation when you leave your desk. Your actions on a validated computer are a legal record. Computerized systems, WHO guidelines. All GMP-related computerized systems should be validated. Appropriate installation and operational qualifications should demonstrate the suitability of the systems to perform the assigned tasks without error. Existing systems that were not validated at the time of installation should be retrospectively validated. Computerized systems should have sufficient controls to prevent unauthorized access or changes to system parameters or data. Written procedures should be available for the operation and maintenance of computerized systems. For manually entered data, there should be secondary checks to ensure the accuracy of the data entered. Incidents related to these systems should be recorded and investigated. Additionally, any changes or repairs should be formally authorized, documented, and recorded, and the systems should be revalidated. Backups of all data and systems parameters are an absolute must. Documentation and records. Document retention. A procedure should be established for retaining all appropriate documents, e.g., development history reports, scale-up reports, technical transfer reports, process validation reports, training records, production records, control records, and distribution records. The retention periods for these documents should be specified. Typically, all production control and distribution records should be retained for at least 1 year after the expiry date of the batch. For APIs with retest dates, records should be retained for at least 3 years after the batch is completely distributed. Many companies maintain their records for longer in the case of litigation or to comply with other non-pharmaceutical regulations. All entries should be making the entry. Corrections should not destroy or make the original unreadable and should be dated and signed. Original copies of all records should be readily available and accessible. Documentation and records. Continued. Batch production records. Master batch records must exist for each intermediate and API and should include complete information relating to the production and control of each batch. When a copy of the batch production record is made for production, it should be checked before issuance to assure that it is the correct version and a legible, accurate reproduction of the master production record. If the batch production record is produced from a separate part of the master document, that document should include a reference to the current master production instruction being used. These records should be numbered with a unique batch or identification number, dated and signed when issued. In continuous production, the product code together with the date and time can serve as the unique identifier until the final number is allocated. Documentation of completion of each significant step in the batch production records should include dates and, when appropriate, times, identifiers of equipment and personnel, specific identification of each batch, actual results for critical process parameters, sampling performed and results, accurate labeling, and any noted deviations. Documentation and records. Continued. Good documentation practice. Documentation for the completion of each significant step in the batch production records, batch production, and control records should include dates and, when appropriate, times, identity of major equipment, e.g. reactors, dryers, mills, specific identification of each batch, including weights, measures, and batch numbers of raw materials, intermediates, or any reprocessed materials used during manufacturing, actual results recorded for critical process parameters, any sampling performed, signatures of the persons performing and directly supervising or checking each critical step in the operation. There should also be a second check signature for all weighings or measurements and additions of materials and reagents and of all transcribed records of critical process parameters in any calculations required during the process. In-process and laboratory test results, actual yield at appropriate phases or times, description of packaging and label for intermediate or API, representative label of API or intermediate if made commercially available, any deviation noted, its evaluation and investigation conducted if appropriate, or reference to that investigation if stored separately, results of release testing. Documentation and records. Continued. Laboratory control records. Laboratory records are critical records for the release of a product for distribution and are therefore subject to the same GMP rigor and document control as product batch records. Laboratory records should include complete data derived from all tests conducted to ensure compliance with established specifications and standards, including examinations and assays, as follows. 1. A description of samples received for testing, including the material name or source, batch number or other distinctive code, date sample was taken, storage conditions, and where appropriate, the quantity and date the sample was received for testing. 2. A statement of or reference to each test method used. 3. A statement of the weight or measure of sample used for each test as described by the method, and data on cross-referenced to the preparation and testing of reference standards, reagents, and standard solutions. 4. A complete record of all raw data generated during each test, in addition to graphs, charts, and spectra from laboratory instrumentation properly identified to show the specific material and batch tested. 5. A record of all calculations performed in connection with the test, including, for example, units of measure, conversion factors, and equivalency factors. 6. A statement of the test results and how they compare with established acceptance criteria. 7. The signature of the person who performed each test and the dates the tests were performed, along with the date and signature of a second person showing that the original records have been reviewed for accuracy, completeness, and compliance with established standards. Documentation and records. Continued. Analytical methods and OOS. Complete records should also be maintained for any modification to established analytical methods, periodic calibration of laboratory instruments, apparatuses, gauges, and recording devices, all stability testing performed on APIs, out of specification OOS investigations. Documentation and records. Continued. Records. Records of equipment use, raw materials, intermediates, API labeling, and packaging materials form part of the master production instructions. These must be accurate and kept up to date. Batch production records are the legal record of any production batch, and they need to be completed accurately at the time of manufacture. Signatures are required on these documents for a legal record. Documentation and records. Continued. Batch record review. Batch production and laboratory records of critical process steps must be reviewed and approved by the quality units before an API is released for supply. Written procedures should be established and followed for the review and approval of batch production and laboratory control records. Batch production and laboratory control records of critical process steps should be reviewed and approved by the quality unit before an API batch is released or distributed. All deviation, investigation, and OOS reports should be reviewed as part of the batch record review before the batch is released. Material management. Material management. There should be written procedures describing the receipt, identification, quarantine, storage, handling, sampling, testing, and approval or rejection of materials and the evaluation of suppliers of critical materials. If the supplier of a critical material is not the manufacturer, the name and address of that manufacturer should be known. Receipt. Upon receipt and before acceptance, each container or grouping of containers of materials should be examined visually for correct labeling, including correlation between the name used by the supplier and the in-house name if these are different, container damage, broken seals, and evidence of tampering or contamination. Quarantine. Materials should be held under quarantine until they have been sampled, examined, or tested as appropriate and released for use. Cross-contamination. There should be assurance that bulk deliveries in non-dedicated tankers are not cross-contaminated. Labeling. Each container or grouping of containers, batches, of materials should be assigned and identified with a distinctive code, batch, or receipt number. This number should be used in recording the disposition of each batch. A system should be in place to identify the status of each batch. Production and in-process controls. Control of critical activities. Written procedures should be established to monitor the progress and control the performance of processing steps that cause variability in the quality characteristics of intermediates and APIs. Acceptance criteria should be defined based on pre-existing empirical data. Critical in-process controls and critical process monitoring, including the control points and methods, should be stated in writing and approved by the quality unit. In-process controls can be performed by qualified production department personnel using validated methods. The process can be adjusted without quality unit approval if the adjustments are made with pre-established limits. Production and in-process controls. Continued. Actual yields. Actual yields should be compared with expected yields at designated steps in the production process. Expected yields with appropriate ranges should be established based on previous laboratory, pilot scale, or manufacturing data. Production and in-process controls. Continued. Deviations. All deviations should be documented and explained, and critical deviations should be investigated. Deviations in yield associated with critical process steps should be investigated to determine their impact or potential impact on the resulting quality of the affected batches. Production and in-process controls. Continued. Processing status. The processing status of major units of equipment should be indicated either on the individual units of equipment or by appropriate documentation, computer control systems, or alternative means. Materials to be reprocessed or reworked should be appropriately controlled to prevent unauthorized use. Production and in-process controls. Continued. In-process sampling and control. The acceptance criteria and type and extent of testing can depend on the nature of the intermediate or API being manufactured, the reaction or process step being conducted, and the degree to which the process introduces variability in the product's quality. Less stringent in-process controls may be appropriate in early processing steps, whereas tighter controls may be appropriate for later processing steps. In-process sampling should be conducted using procedures designed to prevent contamination of the sampled material and other intermediates or APIs with well-defined upper and lower limits. Procedures should be established to ensure the integrity of samples after collection. OOS investigations are not normally needed for the in-process tests that are performed for the purpose of monitoring and/or adjusting the process. Production and in-process controls. Continued. Blending batches. Blending is the process of combining materials that have the same specification to produce a homogeneous intermediate or API. In-process mixing of fractions from single batches or combining fractions from several batches for further processing is considered to be part of the production process and is not considered to be blending. Out of specification, OOS batches should never be blended to meet specification. Blending of small batches to increase batch size or blending of tailings from batches of the same intermediates or API form a single batch. Blending processes should be adequately controlled and documented, and the blended batch should be tested to confirm it meets specification. The batch record of the blending process should allow traceability back to the individual batches that make up the blend. Blending processes should be validated for ensuring homogeneity of the blend. Production and in-process controls. Continued. Contamination control. Residual materials can be carried over into successive batches of the same intermediate or API if there is adequate control. Such carryover should not result in the carryover of degradants or microbial contamination that may adversely alter the established impurity profile of the API. Production operations should be conducted in a manner that will prevent contamination of intermediates or APIs by other materials. Precautions to avoid contamination should be taken when APIs are handled after purification. Packaging and labeling. Packaging and labeling of products. There should be written procedures describing the receipt, identification, quarantine, sampling, examination, and/or testing and release and handling of packaging and labeling materials. Packaging and labeling materials should conform to established specifications. Those that do not comply must be rejected. Records should be maintained for each shipment of labels and packaging materials showing receipt, examination, or testing and whether they are accepted or rejected. Packaging and labeling. Continued. Label issuance and control. Access to the label storage areas should be limited to authorized personnel only. Procedures should be used to reconcile the quantities of labels issued, used, and returned, and to evaluate discrepancies found between the number of containers labeled and the number of labels used. Such discrepancies should be investigated, and the investigation should be approved by the quality unit. All excess labels bearing batch numbers, other batch-related printing, or obsolete, outdated labels should be destroyed. Returned labels should be retained and stored in a manner that prevents mix-ups and provides proper identification. Printing devices used to print labels for packaging operations should be controlled to ensure that all imprinting conforms to the print specified in the batch production record. Printed labels issued for a batch should be carefully examined for proper identity and conformity to specifications in the master production record. The results of this examination should be documented. A printed label representative of those used should be included in the batch production record. Packaging and labeling. Continued. Packaging and labeling operations. There should be documented procedures designed to ensure that the correct packaging materials and labels are used. Labeling operations should be designed to prevent mix-ups. They should be physically or spatially separated from operations involving other intermediates or APIs. Packaging and labeling facilities should be inspected immediately before use to ensure that all materials not needed for the next packaging operation have been removed. This examination should be documented in the batch production records, the facility log, or other documentation system. Packaging and labeling. Continued. Labeling. Labels used on containers of intermediates or APIs should indicate the name or identifying code, the batch number of the product, the storage conditions. If the intermediate or API is intended to be transferred outside the control of the manufacturer's material management system, the name and address of the manufacturer, quantity of contents, and special transport conditions, and any special legal requirements should also be included on the label. For intermediates or APIs with an expiry date, this date should be indicated on the label and certificate of analysis. For intermediates or APIs with a retest date, the retest date should be indicated on the label and/or certificate of analysis. Storage and distribution. A. It is key to have storage facilities to maintain materials under appropriate conditions. For example, temperature and humidity are often controlled. Accurate records of these conditions should be maintained if they are critical for the maintenance of material characteristics. B. Unless there is an alternative system to prevent the unintentional or unauthorized use of quarantined, rejected, returned, or recalled materials, separate storage areas should be assigned for their temporary storage until the decision as to their future use has been taken. C. APIs and intermediates should only be released for distribution to third parties after they have been released by the quality unit. APIs and intermediates can be transferred under quarantine to another unit under the company's control when authorized by the quality units and if appropriate controls and documentation are in place. Products should be transported in a manner that does not affect their quality. Laboratory controls. Specifications. All specifications, sampling plans, and test procedures should be scientifically sound and appropriate to ensure that raw materials, intermediates, APIs, labels, and packaging materials conform to established standards of quality and/or purity. Specifications and test procedures should be consistent with those included in the registration/filing. There can be specifications in addition to those in the registration/filing. Specifications, sampling plans, and test procedures, including changes to them, should be drafted by the appropriate organizational unit and reviewed and approved by the quality unit. Appropriate specifications should be established for APIs in accordance with accepted standards and be consistent with the manufacturing process. The specifications should include a control of the impurities. If the API has a specification for microbiological purity, appropriate action limits for total microbial counts and objectionable organisms should be established and met. If the API has a specification for endotoxins, appropriate action limits should be established and met. Laboratory controls. Continued. OOS. Any OOS result obtained should be investigated and documented according to a procedure. This procedure should require analysis of the data, assessment of whether a significant problem exists, allocation of the tasks for corrective actions and conclusions. Any resampling and/or retesting after OOS results should be performed according to a documented procedure. Laboratory controls. Continued. Labeling. Reagents and standard solutions should be prepared and labeled. Use by dates should be applied as appropriate for analytical reagents or standard solutions. Laboratory controls. Continued. Primary reference standards. Primary reference standards should be used as appropriate. The source of each primary reference standard should be documented, and these records should be maintained for each primary reference standard storage and use in accordance with the supplier's recommendations. Primary reference standards obtained from an appropriate, officially recognized source are normally used without testing if stored under conditions consistent with the supplier's recommendations. Where a primary reference standard is not available from an officially recognized source, an in-house primary standard should be established. Appropriate testing should be performed to fully establish the identity and purity of the primary reference standard. Laboratory controls. Continued. Secondary reference standards. Secondary reference standards should be appropriately prepared, identified, tested, approved, and stored. The suitability of each batch of secondary reference standard should be determined prior to first use by comparing against a primary reference standard. Each batch of secondary reference standards should be periodically requalified in accordance with a written protocol. Laboratory controls. Continued. Testing of intermediates and APIs. Every batch of intermediate or product should be tested to determine conformance to specifications. An impurity profile describing the identified and unidentified impurities present in a typical batch produced by a specific controlled production process should normally be established for each API. The impurity profile should include the identity or some qualitative analytical designation, e.g., retention time, the range of each impurity observed, and classification of each identified impurity, e.g., inorganic, organic, or solvent. The impurity profile is normally dependent upon the production process and origin of the API. The impurity profile should be compared at appropriate intervals with the impurity profile in the regulatory submission or compared with historical data. Appropriate microbiological tests should be conducted on each batch of intermediate and API where microbial quality is specified. Laboratory controls. Continued. Certificate of analysis. An authentic certificate of analysis, COA, should be issued for each batch on request. Information on the name of the intermediate or API, including where appropriate, its grade, the batch number, and the date of release, should be provided on the COA. For intermediates or APIs with an expiry date, the expiry date should be provided on the label and COA. For intermediates or APIs with a retest date, the retest date should be indicated on the label and/or COA. The certificate should list each test performed in accordance with compendial or customer requirements, including the acceptance limits and the results obtained. Certificates should be dated and signed by authorized personnel from the quality unit and should show the name, address, and telephone number of the original manufacturer. Laboratory controls. Continued. Stability. A documented, ongoing testing program should be designed to monitor the stability characteristics of APIs, and the results should be used to confirm appropriate storage conditions and retest or expiry dates. The test procedures used in stability testing should be validated and be stability indicating. Stability samples should be stored in containers that simulate the market container. The first 3 commercial production batches should be placed on the stability monitoring program to confirm the retest or expiry date. Thereafter, at least 1 batch per year of API manufactured should be added to the stability monitoring program and tested at least annually to confirm the stability. For APIs with short shelf lives, testing should be done more frequently. Laboratory controls. Continued. Expiry and retest dating. When an intermediate is intended to be transferred outside the control of the manufacturer's material management system and an expiry or retest date is assigned, supporting stability information should be available. An API expiry or retest date should be based on an evaluation of data derived from stability studies. Common practice is to use a retest date, not an expiration date. A representative sample should be taken for the purpose of performing a retest. Preliminary API expiry or retest dates can be based on pilot scale batches if the pilot batches employ a method of manufacture and a procedure that simulates the final process to be used on a commercial manufacturing scale. Quality of the API represents the material to be made on a commercial scale. Qualification process. Qualification is usually carried out by conducting the following activities individually or combined. Design qualification, DQ. Documented verification that the proposed design of the facilities, equipment, or systems is suitable for the intended purpose. Installation qualification, IQ. Documented verification that the equipment or systems, as installed or modified, comply with the approved design. Operational qualification, OQ. Documented verification that the equipment or systems, as installed or modified, perform as intended throughout the anticipated operating ranges. Performance qualification, PQ. Documented verification that the equipment and ancillary systems, as connected together, can perform effectively and reproducibly based on the approved process method and specifications. Before process validation activities may start, appropriate qualification of critical equipment and ancillary systems should be completed. Validation. Process validation. The number of process runs requiring validation should depend on the complexity of the process or the magnitude of the process change being considered. For prospective and concurrent validation, 3 consecutive successful production batches should be used as a guide, but there may be situations where additional process runs are warranted to prove consistency of the process. Critical process parameters should be controlled and monitored during process validation studies. Process parameters unrelated to quality, such as variables controlled to minimize energy consumption or equipment use, need not be included in the process validation. Process validation should confirm that the impurity profile for each API is within the limits specified. The impurity profile should be comparable to or better than historical data and, where applicable, the profile determined during process development or for batches used for pivotal clinical and toxicological studies. Validation continued. Periodic review. System and processes should be periodically evaluated to verify that they are still operating in a validated manner. Where no significant changes have been made to the system or process, and a quality review confirms that the system or process is consistently producing material meeting its specifications, there is normally no need for revalidation. Critical process parameters should be controlled and monitored during process validation studies and continuously during manufacturing to ensure intermediates and API are manufactured according to validated processes. Validation continued. Cleaning validation. Cleaning procedures should normally be validated. In general, cleaning validation should be directed to those situations or process steps where contamination or carryover of materials poses the greatest risk to API quality. Validation of cleaning procedures should reflect actual equipment usage patterns. If various APIs or intermediates are manufactured in the same equipment and the equipment is cleaned by the same process, a representative intermediate or API can be selected for cleaning validation. This selection should be based on the solubility and difficulty of cleaning and the calculation of residue limits based on potency, toxicity, and stability. Validated cleaning methods are required in multi-product facilities to avoid cross-contamination and ensure the manufacturing of quality intermediates and API. Validation continued. Cleaning validation continued. The cleaning validation protocol should describe the equipment to be cleaned, procedures, materials, acceptable cleaning levels, parameters to be monitored, and controlled in analytical methods. The protocol should also indicate the type of samples to be obtained and how they are collected and labeled. Sampling should include swabbing, rinsing, or alternative methods as appropriate to detect both insoluble and soluble residues. The sampling methods used should be capable of quantitatively measuring levels of residues remaining on the equipment surfaces after cleaning. Validated analytical methods with the sensitivity to detect residues or microbial or endotoxin contaminants should be used. Validation continued. Validation of analytical methods. Analytical methods should always be validated unless the method employed is included in the relevant pharmacopoeia or other recognized standard reference. The suitability of all testing methods used should nonetheless be verified under actual conditions of use and documented. The degree of analytical validation performed should reflect the purpose of the analysis and the stage of the API production process. Appropriate qualification of analytical equipment should be considered before starting validation of analytical methods. Complete records should be maintained of any modification of a validated analytical method. Such records should include the reason for the modification and appropriate data to verify the modification procedure's results are as accurate and reliable as the established method. Rejection and reuse of material. Rejection. Intermediates and APIs failing to meet established specifications should be identified as such and quarantined. These intermediates or APIs can be reprocessed or reworked as described below. The final disposition of rejected materials should be recorded. Reprocessing. Introducing an intermediate or API, including one that does not conform to standards or specifications, back into the process is generally considered acceptable. Also acceptable is reprocessing by repeating a crystallization step or other appropriate chemical or physical manufacturing steps that are part of the established manufacturing process. However, if such reprocessing is used for a majority of batches, it should be included as part of the standard manufacturing process. Reworking. Batches that have been reworked should be subjected to appropriate evaluation, testing, stability testing if warranted, and documentation to show that the reworked product is of equivalent quality to that produced by the original process. Before a decision is taken to rework batches that do not conform to established standards or specifications, an investigation into the reason for nonconformance should be performed. Rejection and reuse of material continued. Recovery. Recovery of reactants, intermediates, or the API is considered acceptable, provided that approved procedures exist for the recovery and the recovered materials meet specifications suitable for their intended use. Solvents can be recovered and reused in the same processes or in different processes, provided that the recovery procedures are controlled and monitored to ensure that solvents meet appropriate standards before reuse or commingling with other approved materials. Returns. Returned intermediates or APIs should be identified as such and quarantined. If the conditions under which returned intermediates or APIs have been stored or shipped before or during their return, or the condition of their containers casts doubts on their quality, the returned intermediates or APIs should be destroyed as appropriate. Change control. Change control. A formal change control system should be established to evaluate all changes that may affect manufacturing quality. Only approved changes should be implemented. Written procedures. Written procedures should provide for the identification, documentation, appropriate review, and approval of changes in raw materials, specifications, analytical methods, facilities, support systems, equipment including computer hardware, processing steps, labeling and packaging materials, computer software. Implementation. When implementing approved changes, measures should be taken to ensure that all documents affected by the changes are revised. After the change has been implemented, there should be an evaluation of the first batches produced or tested under the change. Notification. Current dosage form manufacturers should be notified of changes from established production and process control procedures that can impact the quality of the API. Contract manufacturers, including laboratories. All contract manufacturers, including laboratories, should comply with GMP defined in this guide. Special consideration should be given to the prevention of cross-contamination and to maintaining traceability. Contract manufacturers, including laboratories, should be evaluated by the contract giver to ensure GMP compliance of the specific operations taking place at the contract sites. There should be a written and approved contract or formal agreement between the contract giver and the contract acceptor that defines in detail the GMP responsibilities, including the quality measures of each party and liability to audits. All agents, brokers, traders, distributors, repackers, and relabelers should comply with GMP as defined in WHO Technical Report Series number 957, 2010, Annex 2. Biologicals. The code provides a specific guidance for APIs manufactured by cell culture or fermentation. Classical fermentation uses microorganisms existing in nature to produce APIs. Biotechnological uses cells or organisms generated or modified by recombinant DNA. Appropriate controls must be in place at all critical stages. Particular attentions must be paid to monitoring organism growth and viability. APIs used in clinical trials. For APIs used in clinical trials, appropriate GMP must be followed. An independent quality unit and quality system must be established and functioning. Control of raw materials, production, and validation systems must be established and functioning. Where API manufacture is for single batches, different mechanisms for GMP compliance may be considered. Case study, cross-contamination. This case study illustrates the importance of facility design in the control of cross-contamination. It involves an API manufacturer operating in an older facility in which the design of the production areas did not meet current GMP expectations. Often in such situations, the regulatory authorities recognize that making significant changes to the facility takes time and money and must be planned. Nevertheless, there is an expectation that the risk will be managed, particularly when it involves the risk of cross-contamination. The problem concerns a drying and packaging room in a multi-product API manufacturing facility. In this area, in particular, the risk of cross-contamination is higher than in reactor rooms and other areas. Drying and packaging involves open handling of large quantities of powder. A large amount of powdered API is often discharged into the room. Operators are required to wear personal protective equipment, PPE, which should be put on in an airlock. The purpose of an airlock is twofold: to allow personnel to change into and out of their PPE and to provide an air barrier to prevent airborne API from escaping out into the circulation corridor and possibly into other rooms in the facility. The potential for this to happen is high, even in the best designed facilities, if the correct pressurization regime is not in place. See below. The API facility in this case did not have any airlock, and the personnel would step from the room into the corridor and then put on their PPE in the room, not recommended. Coming out of the room, personnel would take off and bag their PPE gowns for disposal in the room. The breathing equipment would also be taken off in the room, would be wiped down, stored in the room. Both the gowns and the PPE are at risk of contamination since there is turbulent air in the room. There's also the risk of contamination being blown straight out from the room into the corridor when the door is opened, creating turbulence and reducing the differential pressure to zero. Any contamination in the corridor has a high potential to blow into another production room since the air in the corridor is positive with respect to all production areas. During a customer GMP audit, this problem was identified as a major observation due to the high risk of cross-contamination. The regulatory expectation for the facility design typically involves the airlock being at a negative pressure with respect to the corridor, and the room may be negative with respect to the airlock such that contamination does not find its way out of the room and into the airlock. It also allows for PPE to be removed in the airlock and bagged for disposal, or bagged for transfer to the wash bay, such that contaminated PPE does not contaminate the corridor. Before the PPE is introduced back into the airlock for the next product or the next batch of the same product, the room must be cleaned down between batches and between products. For highly potent APIs, the pressure regime may change slightly such that the airlock is positive to both the room and the corridor, a pressure bubble. This gives even greater protection against the possibility of contamination getting into the corridor. Corrective actions. The root cause of the problem was self-evident, as was the corrective action to redesign the facility such that the drying and packaging room had an airlock. This would be a longer-term plan as it would involve closing down a significant proportion of the facility for a period long enough to construct an airlock, install additional HVAC ducts, and balance the system such that the airlocks and the rooms would be a negative pressure with respect to the corridor. Because there was a reluctance on the part of the company to take this action, it was determined that the situation would be managed by introducing additional procedures. These involved storing the clean PPE in a separate room across the corridor. This room was also used for gowning, and operators would then walk across the corridor into the drying and packaging room. On exiting the room, the PPE is removed. The gowns are removed and bagged to take out for disposal, and the breathing equipment is cleaned and taken back into the room across the corridor for storage. Not every story has a complete ending. This one is still in progress. These measures still involve some residual risk that contamination from the room will get out into the corridor, either on the breathing equipment, on the outside of the bags containing the gowns, or on the clothing of the people coming out of the room due to dust that may settle on the people after they have removed their gowns. A further risk is the loss of differential pressure and creation of turbulence each time that the door to the room is opened during operations and before the room and the equipment have been cleaned down. Some of these residual risks could be managed and cleaning procedures and by minimizing the movement of people in and out of the room during operations, but these measures are not as effective and safe as building an airlock for entry into the room. Case Study: Beta-lactam contamination. This case study provides an example of the importance of evaluating environmental risks when determining the overall facility control strategy. By facility control strategy, we mean the way in which people, materials, consumables enter the site, the way in which utilities are located and controlled, and the way in which facilities maintenance can have a large impact on how environmental contamination is controlled. In this particular case, the problem was not of the company's making, but nevertheless it fell to the company to address the problem or stop manufacturing. It involves an API FPP, finished pharmaceutical product, manufacturer located next to a large beta-lactam manufacturing plant. Without going into how the discovery was made, the company facility started to find traces of beta-lactams in the production areas. The first actions taken were to try to directly address the root cause, which was clearly that the penicillin residues were being pumped into the local air by the company's neighbor. This involved trying to get the local environmental regulators to require the company to stop polluting the air. Unfortunately, in the country where this incident took place, and surprisingly it was in a Western industrialized country, the path to getting the neighbor to stop polluting is a challenging one, and a lot of investigation and evidence is required to prosecute a company in such circumstances. Consequently, the API FPP manufacturer decided that all they could do was to monitor the residues in the facility and in the product and try to control the problem with some engineering controls. This approach was of course not at all acceptable to the regulatory authorities, who considered that the root cause was not being addressed. The method of detection was not adequate to detect residues in the product, and it is not acceptable to test quality into the product. This company received a regulatory observation, and at the follow-up visit, the FDA issued a warning letter. The company was requested to describe how it intended to prevent the ingress of beta-lactam residues into the facility. At this point, the company hired in a team of experts to address the problem. The analytical method chosen was the Delvo test method, see reference 1, which is used to test for penicillin residues in milk, which can detect penicillin reliably down to 0.025 units per milliliter, and less reliably down to 0.0025 units per milliliter. However, the reliability, accuracy, and precision of this method were dramatically reduced when it was used to test for residues in the API and FPP. This method was not intended for such use. The GMP consultant involved had to ensure that, as part of the evaluation of the problem, the team was fully informed regarding the regulatory expectations for control of cross-contamination from penicillins and cephalosporins. These products, both APIs and dosage forms, must be manufactured in totally segregated facilities. This normally means at a dedicated manufacturing site. There are some companies that manufacture beta-lactam drug products at the same site as other drug products, but normally in a segregated facility, which is normally a separate building. Also, penicillins and cephalosporins should be segregated from each other because cephalosporins are often given to patients who are sensitive to penicillins, although the incidence of cross-sensitivity is increasing, and often the prescriber will look for an antibiotic that is not in either class of products. Whether the facility is in the same building or a separate building, the segregation should include totally separate equipment, manufacturing and support areas, and separate HVAC utilities. There must be very strict controls for personnel. Typically, personnel who have worked in the beta-lactam facility cannot enter other manufacturing or support areas on the same day and must shower and wear different clean street clothes even to enter the changing area. This particular issue for QA, QC, validation and maintenance personnel and contractors who may be required to enter both the penicillin or cephalosporin manufacturing facility and other facilities in the course of a week. There must also be strict controls on incoming materials, and a separate warehouse is preferred. For cleaning, there should be no detectable residues, and the generally accepted limit of detection is less than 10 ppb. It is worthy of note at this stage that the API FPP manufacturer was a biologicals manufacturer, but this would have little bearing on the problem and could quite easily have occurred at a synthetic API FPP plant. Perhaps the risk of cross-contamination would be even more likely if it is considered that biologicals facilities operate their processes in classified clean rooms where access, gowning, movement of people and materials is generally more strict controlled than in synthetic API facilities. The expert team consisted of a GMP consultant, an HVAC consultant, and an API expert. Working with the company's team assembled to address this problem, a full evaluation was performed. This took form of a fishbone analysis of the potential sources of ingress of penicillin residues. This was followed by a failure modes and effects analysis, FMEA, of each potential source to determine what might have occurred to allow the residues to enter the facility and how the failure risk could be mitigated. The FMEA was quantitative, and this was an important factor because it was clear from the outset that some potential sources were far more likely than others to allow ingress. The fishbone analysis was also very revealing because during the evaluation of the potential sources, a number of other GMP concerns were identified, which were previously unknown to the site management, and these also had to be addressed. There were two surprises in terms of potential sources. One, the employees of the two companies often interacted with each other in a nearby cafeteria at lunchtime. It was discovered that some of the company employees wore their factory scrubs during the lunch break, since there was no requirement to remove them and change back into street clothes for the lunch break. 2. Employees of the beta-lactam manufacturer did not change into factory scrubs and were allowed to wear their street clothes under their factory clothing, which, for many of the employees, were lab coats. 3. The two companies used the same laundering service for their factory clothes. This immediately resulted in the suspicion that residues were being introduced into the API facility by the employees on their factory scrubs and by the laundering company, which might be causing cross-contamination if their procedures were not appropriate to prevent it. The laundering company had never been audited. It was determined that the most likely source of the residues was from the off-gas on the fermenters, which was throwing spores and mycelia into the surrounding air. However, other potential sources were the HVAC exhaust from the packaging and encapsulation rooms in the beta-lactam factory. It was impossible to get a definitive evaluation of the level of risk that each of these sources was presenting, since the beta-lactam manufacturing was not prepared to cooperate with the API FPP company, and they were not subject to FDA inspection at that time. The main sources of beta-lactam ingress was determined to be the air intake of the HVAC and receiving of raw materials and consumables. There was considerable resistance to both of these suggestions from within the company. In particular, it was thought highly unlikely that the residues could penetrate the HEPA filters in the HVAC ductwork. Therefore, a small study was undertaken by sampling the ducts of the HVAC system upstream and downstream of the HEPA filters. The filters were taking out a significant proportion of the beta-lactam residues, but not all residues. Residues were found in the ductwork supplying the grade D clean room where the bioreactor was housed and the support areas. There were even residues found downstream of the filters supplying the grade D air. It cannot be assumed that because there are HEPA filters, nothing can get through them. HEPA filters are rated based on removing a high percentage of particles above a certain size. They are not rated for particles below that size, and if there are a sufficient number of particles of the size for which they are rated, a small proportion of those particles will penetrate. The problem had been present for some time, and during that time, the HVAC system would have taken in a large number of beta-lactam particles. Furthermore, the HVAC air intake grills were directly facing the beta-lactam factory, and the predominant wind was from the beta-lactam facility towards the API FPP facility. Other unexpected sources were as follows. 1. Procedures dictated that personnel could enter the building via 3 separate doors. Production and QC staff were to enter by one door, office staff were to enter via another door, and facilities maintenance were to enter by a third door. On a tour around the outside of the site, it was discovered that there were 9 different doors through which people could enter. These were all unlocked during the day, and 2 were found left wide open during the tour. It was also discovered that some production staff would routinely enter via the warehouse, and that there were no controls on the entry of contractor and maintenance staff or their movement around the building. 2. Utilities were housed in a plant area that was ventilated via slatted windows that did not have any glass, only wire grills. Beta-lactam residues were found in the compressed air feed and around the purified water and WFI storage units. These were functionally closed systems, but they were opened up for maintenance and periodic inspections. There was no formal flushing procedure following maintenance. Some water was run away to waste, but it was not proceduralized. 3. The local reservoir supplying potable water was within a few miles and downwind of the penicillin plant. It was not established whether there was a risk to the potable water supply. This was outside the remit of the company. Should this have been reported to the government environmental agency? 4. Some of the consumables received in the warehouse were transferred to production in their original outer packaging. Corrective actions. 1. Awareness training was provided to all staff to appraise them of the problem and of the proposed corrective actions, and to get their commitment to implementing these actions. 2. In order to prevent the main source of ingress, the HVAC system, it was necessary to remove all contaminated ductwork and replace it. The contaminated HEPAs were replaced, and an additional bank of HEPAs were installed immediately after each air handling unit to reduce the burden on the final HEPA filters supplying air to the room. 3. The air intakes were relocated such that they were away from and not facing the beta-lactam factory. 4. A new entry lobby into the building was constructed, and entry into the office was strictly segregated from the production areas. Office staff enter by one door, and entry to the production areas is via a single entry into changing rooms where street clothes are removed and factory scrubs are put on. 5. Movement between office areas, the maintenance workshop, and production areas was strictly controlled, as was the movement of materials and equipment. Maintenance staff had to keep their tools in boxes which did not leave the production areas. 6. An airlock was constructed for the goods receiving area, and all materials were received in outer plastic wraps, which were wiped down before taken into the warehouse, or were removed from their outer containers. All outer containers and plastic wraps were removed prior to materials being transferred to production. 7. The plant room windows were closed up, and the plant room was provided with a filtered air supply. Corrective actions continued. 1. The flushing of the water storage vessels following maintenance was proceduralized, and the pharmaceutical-grade water was tested for beta-lactam residues on a regular basis. No residues were detected. The incoming potable water supply was also sampled for penicillin residues, and none were detected. 2. Production was stopped while work on the HVAC system was performed, and as an interim measure while other construction work was being performed. New, more selective beta-lactam assays were developed which were capable of detecting beta-lactam residues down to 10 parts per million in the product. All batches of product were routinely tested, as were in-process samples at various steps in the process. 3. A cafeteria was built on-site for staff such that they did not have to share lunch facilities with the beta-lactam company employees. 4. A significant level of staff training was undertaken to ensure that the new measures and procedures were understood. 5. The laundry service was audited, and as a result, an alternative service was provided by the same company, dedicated washing machines and handling of clean laundry. One of the most difficult challenges was to convince the company that some of the potential sources needed further investigation and should be taken more seriously. Once the residue data from the HVAC, the plant room, and warehouse were made available, the attitude changed dramatically and the culture within the company also changed. The modifications to the HVAC were completed within a 6-month period, but the other modifications took over 2 years to complete. Prior to the modifications, residues were detected in a number of production areas, in the plant room and in the warehouse, but no residues were ever detected in the product either prior to or following the corrective measures. Therefore, while the residues were getting into the facility, there was no evidence that they were ever getting into the product. Nevertheless, the company could not have justified simply a program of testing. At the next FDA inspection, the warning letter was lifted and the company has had a good record of compliance since that time. The evaluation exercise raised many issues that were unknown to senior management, and this set off a chain of events that led to a change in the culture. This change has been seen as a huge benefit to the company for many years. Some of the measures taken may have been excessive, but because of the lack of positive action initially, the company were forced into a situation where they had to demonstrate that they had left no stone unturned. Final assessment. Review the module before commencing your final assessment. All questions relate directly to information and principles covered in the slides. You can only attempt each question once. Question types may include true-false, drag and drop, and multiple choice. Good luck.